welcome to the first episode of Larson in the Lab. Today I'm going to be going over some different lab equipment that you might use if you get to do a biology lab. So first things first, like I said, we're talking about biology lab equipment, and I'm wearing two very important pieces of equipment. I've got my safety goggles. I've also got my lab apron. I wear my safety goggles to protect my eyes and my lab apron to protect my clothes and my body. These are very important pieces of lab equipment to maintain your safety. So lab apron for my clothes, safety goggles for my eyes. All right, next up, let's talk about some different tools that you might use here. We've got first test tubes. You're probably familiar with test tubes, but they're used to hold small amounts of liquid or other materials. Now, if you set these test tubes down on the countertop, they might roll away. So instead, you're going to set them into a test tube rack, just like this. This can hold multiple different test tubes, and test tube wraps come in all different shapes and sizes. They can be plastic, they can be wooden, and they can hold just like 6 test tubes, or they can hold very many test tubes. Now, if you are also using a test tube and you're wanting to keep something in that test tube for an extended period of time, you might use some rubber stoppers. They look like this. For those test tubes that you just saw, that you would probably use one of these smaller rubber stoppers. These can go into the test tube and seal it so that nothing can escape or get in. If you're using a test tube and you're going to heat it up, you're going to want to hold it using test tube tongs. I'm now showing you test tube tongs holding a test tube. Now, if you're heating a substance, you want to hold the tip of the test tube away from you and your lab partners. If you hold it close to you or your lab partners, material could end up splashing out and burning somebody. That would be really terrible. We don't want to do that. Let's see what else we have. So you also have beakers, which can hold larger amounts of liquid than test tubes, and they're usually measured in increments of 10 or 25, sometimes even 50 milliliters. This is a pretty small one, but there are larger ones you would see in the lab setting. If you want to measure liquid amounts that more precisely than using a beaker, you're going to use graduated cylinders. You're probably pretty familiar with graduated cylinders. You might have used them in different science classes. They come in all different shapes and sizes. When you are heating a beaker, I forgot to mention, if you were heating this beaker up, you would definitely not want to touch it with your hands. Instead, you would use beaker tongs. Notice they have rubber on the ends to help with the insulation of the heat, and beaker tongs can be used to pick up a beaker that's hot. Side note, if glassware is hot, it doesn't look any different than if it's cool. So if you're in a lab that uses heat, always assume that it is hot. So if you want to measure how hot, you can use a thermometer. In the lab, we have these really long thermometers, and they actually have a little rubber piece at the top to make sure that they don't roll off the lab table. But you got to be careful when you're using these. You definitely don't want to break it. These don't have mercury in them, but the older ones might have mercury in them. Next up, let's see. If you're measuring small amounts of liquid, very tiny amounts, you're going to use a dropping pipette. It holds possibly like a millimeter of liquid. We would use it in our water, properties of water lab. If you're using material, if you're heating liquid, or if you're swirling liquid and you don't want it to spill, you would use an Erlenmeyer flask. The edges of it are aimed inward or pointed inward, and that would prevent spills. Erlenmeyer flask. Long word, not that hard to say, Erlenmeyer. All right, let's see what's next. If you are cleaning some of your glassware, or if you need to use a small amount of distilled water, you can use a plastic water squirt bottle. It squirts out the liquid just like that. Though this is not a toy, this is not a squirt gun for using in the classroom. If you're working with evaporation and you want to see what's left behind when a liquid evaporates, you can use an evaporating dish. You can use this with heat to evaporate liquid and see what remains. You might also use a watch glass. Now, you're probably not going to use heat with the watch glass, but you can let something evaporate and then see what remains in the watch glass here. I know it looks like a giant contact lens. It's not. It's a watch glass. All right, next up, how would you move material that is liquid from a large beaker into a smaller beaker without spilling? You would use a funnel. You're probably familiar with these. You might have even seen one in your kitchen. If you are measuring out small amounts of solid material, you might use a scoopula, also known as a spatula, and They come in different shapes and sizes, and they can help you collect small amounts of solid material. How much material did you get? Well, you might use an electronic balance to find out. This is what they look like. They plug into the wall, and they'll give you a mass in grams is the unit that you would want to use. All right. Next up, I've got for you, if you're trying to blend together or mix together two different materials, you might use this glass stirring rod. This is solid glass stirring rod. You want to be careful if you have this on your lab table that it doesn't roll off and break. They are breakable. So just be careful if you are using a glass stirring rod. Next up, I want to talk about collecting samples and possibly growing them. If you're collecting samples of bacteria or fungus, you might use a Petri dish. And an inoculating loop can put some of the material onto the liquid agar inside a Petri dish, and then that bacteria or fungus could grow, and you can watch the growth within the Petri dish. All right, if you're trying to clean any of these materials that I've just shown you, you might use a test tube brush. If it's a test tube or a graduated cylinder, maybe even an Erlenmeyer flask. If you are trying to clean something larger like a beaker, you can use a beaker brush. It's a lot larger than a test tube brush. All right, next up. Last thing about some of these materials, if you are using them to heat a substance, you might use a hot plate to heat up your substance. Now this plugs into the wall. You can choose which level of heat you want to use. It's a hot plate and it is used for heating materials in the lab. We might use this when we do an enzyme lab to talk about how heat impacts enzymes. All right, most of you, when you think of biology, you might think of using a microscope. Now, this is a compound light microscope. You can tell because it has a bunch of different lenses. It has the objective lenses down here that are close to the object, and it has the ocular lens up at the top. Before we start talking about cells later on, we'll talk more about the parts of these microscopes and how to use them. But that is referred to as a compound light microscope. The other type of microscope you might have seen in a biology classroom is called a stereoscope. This is a stereoscope. Notice when I carry these microscopes, I'm using both hands, holding the bottom and then supporting the top by holding the arm. This is called the arm, and this is called the base. This is not have the same type of compound lenses that the other microscope had. This actually has two ocular lenses and then one larger objective lens, which you can rotate for different levels of magnification. This is used for looking at specimens that you have dissected. It's called a stereoscope. All right, the last thing you might think of when you think of biology or think back to maybe 7th grade science is dissection. I think in 7th grade you probably dissected a frog, and most likely you used a pan like this. This is called a dissecting pan, and you would put your specimen in here as you dissect it because some liquid might come out and you don't want that all over the lab table. This is also a rubbery material, so you can use pens to pin down different parts of whatever you're dissecting. When you're dissecting, you might use a dissecting toolkit. This is going to be full of different tools like forceps and scissors and pens. You can see in there different types of tools for dissection. The last thing you might use when dissecting are forceps. These can be used in dissection, but they can also be used in other labs when you're picking up smaller objects. If you were to dissect something or collect some sort of sample that you wanted to look at under the microscope, you would need a glass microscope slide. This is a slide, it's a larger rectangle shape. And if you were making your own slide, you would use what we call a cover slip. Now a cover slip it comes in a little box like this, and that's a small piece of plastic. It's a tiny little square of plastic. So if you're using your own microscope slide and you're putting your cover slip on, you would put your specimen on the microscope slide. You'd put a little squirt of water, and then you would place this just like this, starting from the edge and going downward, pressing downward to eliminate any air bubbles. Air bubbles will make it hard to see under the microscope. That's all for today. Thanks so much for watching Larson in the Lab.